Hello, this is Professor White, and welcome to Biology 2, which is your laboratory in biology. So, today I will be explaining to you what we will be learning for this week one of our laboratory in biology. So, we'll start first with what it is. So, our week one topic is about acids and bases. And the learning objectives for our lab is to measure the acidic, basic, and neutral solutions and collect data. Additionally, we're going to examine the factors that impact the pH of a solution, as well as to demonstrate how to adjust the pH of a solution and demonstrate how to maintain the pH using buffer solution. And lastly, to discuss the differences in the pH of different household solution. But we'll start first with the importance of pH. Well, our life is pretty much dependent on pH, as you have to say, mainly because the hydrogen concentration of every solution in your life, um, whether in your own blood or those drinks that we drink, like soda or coffee, one way or another, it is affected by the hydrogen concentration of a solution or its pH. Now, it determines the suitability of that solution to perform various tasks, such as, let's say, for example, for cleaning solutions at home, such as ammonia, which has a very high pH, while white vinegar, for example, has a low pH. And we know very well that some common substances we use at home has a certain level of pH. But we'll start first with what acids and bases are. So acids normally donates hydrogen ions during chemical reactions. And we can represent an acid as a hydrogen containing compound and we label it as HA. Now, in solution, these compounds tend to dissociate or kind of like break down into ions, wherein the HA or your hydrogen acid basically dissociate into H plus and A minus. Now, the A minus is known as the conjugate base of an acid. Now, the dissociation, let's say, for example, in this particular acid, which is your hydrochloric acid, dissociates into H plus and Cl minus. So this is a very common example of an acid. Now, how about the bases? Now, the bases accept hydrogen ions H plus during chemical reactions. Now, this is represented by letter B. This compound actually tends to accept the hydrogen ions H plus. So in this particular equation, H plus, it accepts that H plus with the acid, which is B, to form Hb plus. Now, the Hb plus ion is known as the conjugate acid of the base. Now, the reaction of ammonia, for example, in this example, in the presence of hydrogen plus ion, will basically form NH4 plus in this example of a base, which is your ammonia. Now, water can actually dissociate into ions. So H2O, we already know that that's the chemical formula for water. It actually breaks down into H plus and OH minus. Now, in pure water, this reaction produces equal quantities of hydrogen ions and hydroxide ions like acid and base. Now, the right and left arrow in here indicates that the reaction can proceed forward or backward reaction. However, the reaction indicated by the left directed arrow is strongly favored, and therefore only small quantities of hydrogen and hydroxide ions are found in water. Now let's now go to the pH scale. So the concentration is a measure of the amount of substance contained per unit volume. Now, one commonly used unit in the amount of a substance is the mole. Now, when used along with a metric unit of volume, you can express the concentration as moles per liter. Now, let's take a look at the range observed in the concentration of ions in solution. And it's very, very large. And therefore, we use what we call a pH scale. Because of that large range, it is ideal to express that hydrogen ions on a scale based on a power of 10. So that's why we have this pH scale. Now, on the pH scale, every unit increase in pH is directly related to the factor of 10 and decreases the hydrogen concentration from left to right. Now, the approximate hydrogen ion concentration and pH for various solution is shown right here. So let's take a look at from left side. You can see that the battery acid has 10 to the 0 hydrogen ion, and therefore it has a pH of 0. Now, as you move to the right, the vinegar has 10 to the negative 2. If you are curious what negative 2 means, it's basically just moving the decimal point to the left-hand side of your 1. So 0.0012.001. So that's the hydrogen concentration of vinegar, which has a pH of 2. And as you go further, tomato juice is 10 to the negative 4, which is pH of 4. Milk is 10 to the negative 6, which is pH 6. Water is 7. And FYI, the 7 is actually a neutral solution. So that's your pure water. Seawater is about 10 to the negative 8, which is the same as pH 8. And as you go further to the right side, which we can see here, the drain cleaner is 10 to the negative 14, which is a basic solution. So right after the neutral solution, that's when your substance become basic. Now, how do we measure pH in solutions? So either we can use, there are actually different ways. You can either use a pH paper, a litmus paper, or pH meters. Now, in our experiment, you will measure the pH of various household solutions, biological fluids, and buffer solutions. So an example of measurement made using the portable pH meter is shown right here. So we're going to use kind of like a pH meter in this simulation. Now let's go to buffer solution. Now in our lab activity, you will get the opportunity to explore how to maintain the pH of a solution within a specific range. It can be a matter of life and death whenever we are trying to maintain biological fluids in the body within a limited pH range. For example, in the pH of your blood, was to kind of like deviate from its average pH, which is 7.3. Now, the cell structure could actually get affected by this change in pH, and it can be life-threatening. Now, fortunately for us humans and other animals, there is this naturally occurring buffer, which is called bicarbonate. And in fact, I'm sure you guys are very familiar that if the pH level in your stomach is actually basically off, then we buy antacids like your Tums just to balance out the acidity inside your stomach and maintain its correct pH. Now, a buffer solution can be made by adding a weak acid, so an acid that contributes only a small percentage of its available hydrogen ions to the solution, and its conjugate base to water. Now, two examples of weak acids and their conjugate bases and the associated buffer systems are given in this table right here. And we can use a convenient shorthand notation to represent our weak acid plus conjugate base buffer. So HA is a weak acid and A minus is a conjugate base. So basically, when you add, let's say, for example, in this example, the acetic acid is a weak acid, and the conjugate base of it is an acetate ion. So it kind of like if you add, let's say, for example, that acetate ion with the acetic acid, then it balances out the pH. And that is your buffer solution. So that's the kind of like the idea. It's kind of like a balancing the amount of acids and bases. So that is your buffer solution and pH. The concentrations of weak acid and its conjugate base don't have to be the same in a buffer solution. In fact, by choosing different relative concentrations, buffers with different pH can be constructed from the same weak acid conjugate base pair. Now, how does the buffer solution help maintain pH? Well, consider the situation here on this figure. Now, on the far left-hand side, we have the buffer solution that consists of HA, which is a weak acid, and its conjugate base, which is A minus. Now, the solution will have a specific pH value that depends on the particular acid-base pair used for the buffer and their relative concentration. Now, in the center diagram, a small amount of acid indicated by the H plus ions is added to the buffer solution. Now, how will this buffer respond? Well, the net result is shown to your right-hand side. And notice that most of the added acid has reacted with the conjugate base A minus, which is the purple one. Now, notice that most of the acid has reacted with the conjugate base A minus in the solution to form more weak acid. So basically, in this illustration, only one of the three added hydrogen ions remains free in the solution and contributes to a change in the solution pH. It is in this manner that a buffer system responds in addition to, acids and maintains its pH. Note that a buffer system can be overwhelmed by the addition of too much acid. Basically, a buffer system will lose its ability to maintain the pH once all of the conjugate bases has been consumed by reacting with added acid. So the buffer system here shown responding to addition of hydrogen plus solution. Basically, how does it work? Well, when there's too much acid right there, you add the base and react. So kind of like add more acid, add base, and then after a while, it forms water and releases the conjugate base. And finally, it should be noted that buffer systems can also be constructed from a weak base and its conjugate acid. Now, the principle and the actions are similar to what we have seen right here. Now, in your lab today, you are going to perform the pH testing of household and biological solution and use that information correctly to classify solutions as acids or bases. So that's the first part. Now, the second part, which is procedures 3 and 4, you will compare the pH changes of a buffer solution and water when acid or base is added to each solution. As you can see, buffer solutions kind of like maintain the pH, right? So that's the idea of the second part of your lab. And last but not least, adding an acid or base to a solution causes changes in the pH of a solution. So you're going to be calculating the percent change of pH, which allows us to understand how large or maybe how small a specific pH change is. And the percent change can either be positive or negative. Now, a positive result indicates that an increase, and a negative result indicates a decrease. I hope that our direction for our lab for this week is clear. And if you need more support or understanding, you can either replay this video or you can send me a message to help you through the week. Again, this is Professor White. Have a great day. Bye.